There we go. So, so a couple of things to start off with. First of all, I want to welcome you and I really appreciate you volunteering for this job. Um, it's a pretty fun aspect of the fungus fair to be a part of the identification process. Um, and hopefully you'll learn a bit and come back next year and help us out with that as well. I wanted to go through the process in general first to give you an idea of what's going to be happening and where you will fit in. So we'll start off by people bringing mushrooms in from forays. So there's half a dozen sort of pre-scheduled forays that are out there. A number of them in the San Mateo County parks. There's an East Bay group. There's a Point Reyes group, and so on. So those folks will go out in the morning and collect into the early afternoon on Saturday, the 7th. And then they'll bring their boxes and bags full of mushrooms to the fungus fair, hopefully more or less pre-sorted. So in other words, they won't have mixed collections. So they'll have uh, wax paper bags, which is the best way to to store mushrooms short term. So they'll have for each wax paper bag, they'll have just one species in each bag. Um, and they'll bring those to the fungus fair, and then they will put those collections on styrofoam trays. Most of them are styrofoam. We have some cardboard ones as well. Say about this big. Some are a little bigger, some a little smaller. And they'll put those out on the trays, and they will put a label with the foray name on the label. And those will be pre-printed for the set forays. So, for example, I'm leading a foray to San Pedro Valley County Park in Pacifica, and so I would come in with my collections, and I would grab a stack of the tags that say that on the say San Pedro Valley County Park on the tag. Put my collections on the trays, put a tag on it, and then set it aside. And so we may ask you to help with that. So you want to make sure that you have just one species per tray, and then one tag for each tray that has the proper foray name on it. And by the way, there are likely to be some sort of unofficial forays that'll come in without without pre-printed tags. So we'll have some blanks that people can just write on there the name of the foray that they did. So, for example, let's say somebody lived over by McLaren Park in San Francisco, and they went out in the morning and collected a bunch of things and brought them back to the fungus fair. Then we can write on there McLaren Park, something like that. And uh, so depending on when you're there and what's needed, again, we may ask you to help with that to sort that out. And by the way, it's okay to just in general, both specifically within a foray and in general, it's okay to consolidate the species. So let's say, for example, that a foray comes in and they have several bags of Amanita muscaria, the red one with the white spots. And so it's ideally all of those are going to go on the same large tray or small box or something like that. Um, it's also okay to consolidate between forays as long as both tags, or if there's three forays, three tags or whatever, as long as all of the tags are on the tray with the collections, right? So let's say that again you have a bunch of Amanita muscarias. You can put them all in one tray or box and make sure all of the tags are associated with those forays are in there. So let me speak to why that's important. Uh, the There are some places, all of the San Mateo County parks, for example, that ask us for species lists at the end of the fungus fair. So if we don't have the tags, uh, then we don't know where they came from, and so we want to make sure that that's the case. That's one reason. The other reason is that we've been keeping track of this for 50 years, more or less. I think there's some lost years in there, COVID related, for example. But there's we've been keeping a lot of data, and who knows? Someday someone might be able to make good use of that data to look at trends, population trends for certain mushrooms, for example, um, or something even more complicated, looking at, say, weather factors related to abundance and things like that. So, so anyway, that's really important to keep the tags with the proper foray name with the collections, right? So, so then imagine that there will be some tables that have sort of a mishmash of different species with the trays. And so one of the things that we're going to ask you to do is to do a pre-sort. There will be a space. So there, I think we've got 5 tables or something like that, where people will be bringing in their collections. They'll be putting them out on these tables. And then you will be taking those collections and putting them on a second set of tables. We have 6 tables. I have 15 categories. And that's what we're going to go over tonight. So don't concern yourself with that quite yet. So we'll have 15 categories, and you will take the trays from the tables that where the forays have set them out, and you will take them and put them in the appropriate place on the second set of tables. That's the pre-sort. Then there will be a gap, and there will be another set of tables where our identifiers will be sitting. And those are the folks who have the expertise to actually put names on these species, uh, both genus and species for each of those collections. Um, and they will be sitting together so that they can communicate with each other. You know, hey, what does this look like to you? How does this smell? Do you think this is such and such, and so on? So they'll be able to talk to each other, and they'll have their reference books and things like that with them, and so on. So it's going to be a lot easier for them to sort of concentrate maybe on a particular genus or a family at a time. Um, and so they can go out and they can find the appropriate place where you have laid the pre-sorted fungi, and so they can just bring them out instead of looking around on, a dozen tables or something like that, looking for that, that other puffball or that other amanita or something like that. So that's going to be really different than what we did last year if you were there, um, and much more organized and concise. Um, Then the collections will be recorded, and there are a couple, 2 or 3 people that will be putting these uh, essentially a foray designation for each species and where it was found, and that's where those tags will come in handy specifically. Uh, then we'll have essentially runners, people who will take those identified and recorded collections, they'll take them out to the display tables. Now, if you've been to a fungus fair before, that should sound familiar. So whether it's our MSSF fungus fair or Santa Cruz or Marin County, they all pretty much do it the same way, where there's a set of tables um, that are arranged in some fashion, maybe taxonomically or maybe by category, which is what we're going to be doing, sort of form and um, other characteristics. Um, and we put those out. We'll have these trays that we can put toothpicks and um, paper like bamboo sticks and label pins into the tray. It's basically a foam, um, and the tray will be covered with oak leaves, so it'll look nice. And then we're going to have a bunch of people who are going to help us to essentially set them up so they look nice. So for example, you'll have, let's say you have 10 of a particular species. So maybe three or four, you'll use toothpicks or the bamboo sticks, the skewers to set a couple of them up as if they were growing up out of the oak duff, oak leaves. And then you'll have some that are laying down, and those will be by category. And uh, what we do is we have um, several tables, about a little more than a dozen tables that are set up in a rectangle, a narrow rectangle. And then when people come to the fair, we'll have uh, um, people who are interested in talking about the fungi behind the tables, talking to the general public, answering their questions, and so on. Some of the mushrooms will be going to what we call the woodland display. So there will be a essentially a tree growing in the middle of the cafeteria at the high school with a bunch of ferns and shrubs and so on, and a bunch more oak leaf duff. Um, and then people will be putting mushrooms in there as if it was a little patch of the woods growing in the middle of the cafeteria. It's really impressive and really a neat thing. So some of the collections are going to go there, um, and then there will be also be a few mushrooms that will be going to other places. So, for example, we're going to have the crafts department, and I'm not sure what Karen will have going there. She's in charge of that. But for example, she might want to do spore prints or something, so she'll come and get some of those, um, and some other of our uh, uh, eh, some other of our attractions will have um, some mushrooms that we've collected. So, but the main thing, most of them are going to go in this display table. All right. Are there any questions about that process? Okay. All right. Well, let's dig in here. So I'm going to share my screen. And why didn't that work? Huh, there we go. All right. Isn't that fancy? So I want to thank Microsoft Office for having the mushroom template for our little demonstration here. That's the last time that you're going to see that screen. So everything, but I thought it was pretty cool that uh, as soon as I said this basic mushroom identification, and the PowerPoint said, "Oh, you're going to love this." So there it is. All right. Um, Okay, so these are the categories that you will be having utilizing to sort the mushrooms, and we're going to go through each of these categories real quickly, uh, and so don't stress too much. This will be on the exam, but no, I'm kidding. Won't be an exam, um, but you can see that there's about 15 different groups there. And we'll go through, like I say, we'll go through each one. Now, if you sort of scan this screen here, you can see that there's a couple of aspects um, that are highlighted. Uh, First of all, you can see that there's a bunch of spore colors that are involved with the gilled mushrooms. So rusty, orange spored, brown spored, black spored, pink spored, white spored. There are actually even a few other colors, but don't worry about that. These are the main categories. And so, so one of the things that I'll be talking about is how you can figure out what color the spores are for the mushrooms that have gills. Um, if you look under the white spore category, you can see the last three there: um, tricholomatoid, colibioid, marasmoid, and mycinoid. If you see that suffix oid, that means like. So tricholoma-like, colibia-like, marasmius-like, mycena-like. So these are statures, and I'll be going through the different kinds of statures that we can expect to see. Um, and then one thing that you aren't seeing here, but except over here with the agaricus, is gill attachment, and so we'll be talking about that too. So really, there are three main characteristics: spore color, gill attachment, and stature. So this photograph was the overflow table at the Mendo foray. Uh, Several years before the COVID event, but we had so many mushrooms, we just didn't have room inside for them, and it was a nice sunny weekend, so we put them all outside. So imagine that it was your job to sort these into categories, and so in fact, that's pretty much what it's going to be like. So let's talk about spore color first. So again, the main ones are white, brown, black, pink, orange, uh, and to put kind of putting with orange, rusty colors. And again, there's a couple other ones. So ideally, you would be taking spore prints, and that's how spore color has kind of come about as and if you have the time and you really want to figure out what you have, you need to know the spore color. That's with the gilled mushrooms. That's the first part of uh, a keying out process is figuring out what the spore color is. By the way, in this illustration, which I found on PBS Utah, PBS Kids Utah, it's really great. This one up here, this one down here, and this one over here, and this one on the other side. So these four in the corners, those are uh, some sort of a pored mushroom, maybe a Tylopolis or uh, Lexinum or something like that. Uh, these ones in the middle with the brown spores, I think those are probably what we call a crepidotus. But if you look closely, you can see there's a bunch of other ones that are sort of cream colored. And now, I typically, uh, someone who's identifying a mushroom, they would do spore print on a white paper. Oh, by the way, I forgot to tell you exactly how to do that. Essentially, you take a mushroom, cut the stem of it, the mushroom off, and set this on paper. And it really should be white paper so that you can tell what color it is. If you put it on colored paper, it can actually skew your concept of what the color is. Now, that's if you're doing identification. If you're doing art, go for it. Use construction paper or whatever you want. And these cream colored spore prints look really great on black construction paper, for example. Now, you may think to yourself, well, wait a minute, if you're going to put a white spored mushroom on white paper, then how can you tell it's even there, right? That's pretty good camouflage, white spore print on white paper. But you can actually tell by holding up the paper and kind of looking at it obliquely. all of these prints are essentially little piles of spores, either where the tubes of say a bolete or a polypore have dropped them out, or along in between the gills of a gilled mushroom. By the way, as I go along here, um, uh, with, without trying to avoid getting into the weeds too much, by all means, interrupt me if you have a question. Um, and you don't need to wait until the very end to ask questions about this stuff. So, um, okay, all right. Here's a little quiz for you. All right, so we have five mushrooms here. And what I want you to do is I want you to look at these mushrooms and guess the spore color. Okay, I'm going to give you a minute to do that. All right. So we're going to start with this one in the upper left corner here. What color are the spores of this mushroom? And you can unmute and call it out.
Yellowish orange.
Okay. Turns out it's white.
Ah.
Ugh. What? Okay. All right. Let's go to the next one. How about this one? What color spores do you think that is? Somebody else. Another person.
Pink.
What did you say?
Pink.
Yeah, it looks like pink, right? Chocolate brown. That's an agaricus like portobello. Yikes. Okay. Somebody else. This one.
Tattoo? Pink.
Pink. Turns out that they're black spores on that one. You can kind of see that a little bit right there. Black spores. Okay. Somebody else call out this one. Okay, yeah, I can see we're getting a little gun shy here because JR is pulling a little fast one on us. All right, well, I'll tell you, this is a white spored mushroom. This one down here, which actually I didn't know until I figured out what it was, that's pink spored mushroom. And by the way, the pink that that I talk about here, the is called mycological pink. It's really more of a sort of a brownish pink color, but this one has that brownish pink color. So here's the lesson, uh, which is going to be frustrating in a lot of cases. Some of the time, not, I would say not the majority of the time, but some of the time, the gill color does not help you out. Most of the time, it does. So, for example, this one down here is a little bit different than the rest because it starts out that pink color like that, but it's going to turn chocolate brown as it matures. And as the spores mature, it'll start to look more like what you see on a portobello. Um, these other ones, like this is a Hygrophilopsis. This over here is a Tricholoma equestri. It's called man on horseback. This little Entoloma down here, and, and this Gomphidius. All of those have, well, actually, forget the Gomphidius, but these three, the two on the right side and the one in the upper left, those gills are pigmented. It doesn't have anything to do with the spore color. Um, and the Gomphidius, that's as that gets older, you'll see black spores, but it's a real trickery kind of a mushroom. All right, so what do you do? Well, again, most of the time you'll be able to tell based on the spore color, but sometimes that you look for other hints. So, for example, if you look on the stipe, this is a Cortinarius, and Cortinarius have rusty brown spores. So you can see that those spores have been caught in these little fibers that are on the stipe. That's a special kind of what we call a partial veil or an annulus that is cobwebby in nature, and that's the Cortinarius almost always have that. Um, we're going to talk more about Cortinarius later. Um, this one over here on the far right, this is a honey mushroom, which is an edible species. Um, and if you look at some of these caps, which are sort of orange in color, you see that they have this white dusting on those caps. And so if you have a situation where you have a cluster of mushrooms like this, um, look at the ones that are underneath other ones, and you oftentimes will see a spore drop happening as a result of that. And anybody know what that kind of mushroom is right there? It's a shroom, dude.
Oh,
So that's a
psilocybe.
psilocybe.
Mhm.
And you can see that they have, instead of a cobwebby annulus, they have a membranous, and this species has a membranous annulus. So it's literally a ring around the top of the stipe. You can see they all have it, and you can see on the top side of that ring that it is very dark. Turns out that the spore print here um, is a purple, black is how it's described. So it's very dark purple color. If you were to look on the underside, like let's see that one right there, a little bit on this one, you can see that that it's not nearly as dark, and that's because the spores are falling on the top of that annulus, as opposed to on the whole thing. So, all right. So for in practical terms, if you don't know, don't worry about it. Ask me if I'm around or somebody else, one of the identifiers, um, if they know what color the spores are on that particular collection. Um, and if there's nobody around, make a guess. Uh, and I'll be periodically going around and just checking your work. Uh, and if I need to move something, I don't get too miffed about that. So I totally understand if you don't get it right 100% of the time. So again, don't stress out about the spore color. Do your best on that in that regard. All right, then the second thing is gill attachment. And I put over here a little cheat sheet. By the way, these illustrations are from a a treatise that Elza Fellingha, who is one of our uh, um, wonderful identifiers, who won't be there. She's the person who's got another thing going, but she won't be there. But anyway, this is her work, so I want to give her credit for that. Okay, so if you look over here on the lower left, you see there's five categories. So free, adnext, adnate, notched, and decurrent. So this has to do with how the gills are attached to the stem. So let's start with free, and there's only one illustration on here, that's number 18, that is free. And that's where the gills are not attached to the stipe at all. Um, and one way to tell that, and I don't want you to do this with every collection, but you should try this with, uh, well, I'll tell you in a minute what you should try it with. But if you get a chance, um, the way to tell for sure if it's free or not is if you break the stipe off of the cap, and it breaks off like a ball and socket, and there's no little pieces of gill on the part of this that's at the top of the stipe. So, for example, if you look at number 19 and number 22, that may look free, especially as it gets older. So as mushrooms get older, they expand. And even some of these other ones in the middle here, or even like number 23 here, sometimes as they get older and older, those gills literally break right close to the stipe. But you're going to see little pieces of the gills on the top of the stipe, right? So free is going to break like a ball and socket. And there's only a couple uh, of groups that we commonly see in California, that three groups that I can think of that have free gills. So we'll be talking a little bit more about free gills in a minute. Now, adnext and adnate and notch, for that matter, are basically that they're attached somehow. So add next, which is 19, 20, and then jump up here to 22. These are where the gills go across and then up, right? Across and then up. And the attachment is fairly high on the stipe. If you compare that to adnate, which is 21 and 24 over here, you can see that the gill basically goes straight across. That's pretty easy to see compared to adnext versus adnate. Now, like once again, oh, and let me talk about notched real quickly. So, and there's a jargon word for notched, and it's emarginate. But number 23 here is notched. So it's basically where it looks like it's going to go up as if it were add next, like number 19, but it does a little jog and goes straight across, like we see in 21. That's notched. For the most part, you're going to not you will not need to know the difference between add next, adnate, and notched when you're doing these pre-sorts. It's not a real key feature, whereas free might be, and it also decurrent might be an important feature. So 27, 28, and 29 are decurrent. So you see the gill goes across and then it runs down the stipe. So the width of the gill basically increases as you get closer to the stipe, as opposed to decreasing if it's adnext, or staying the same more or less if it's adnate. All right. So just a real quick primer on that. Again, don't worry too much if you get it wrong. Again, I will be going around and checking to make sure. And here's some examples. So here is a member of the genus Lepiota, and you can see that the gills run up, and there's actually a sort of a little collar at the top of the stipe, and where the gills are not attached at all. This is some of you may recognize as a bluet. It's an edible, sought after edible species. Has this beautiful lilac color. And these gills, they look like they're going to be free, right? It looks like they're not attached. But if you were to cut this mushroom right down here and look at it from the side, you would see that either the gills are definitely attached to the stipe, or they were before the cap expanded and basically broke the gills off away from the stipe. So this is an example of adnext. Here's an example of adnate. So here's where the gills come pretty much straight across. The width of the gill doesn't change substantially from a little ways out to the stem. And then here's decurrent. Okay. So once again, basically you should know or have a pretty good idea of what free means, a pretty good idea of what decurrent is, and then everything else is in between. All right. Okay, don't hesitate to ask any questions as I go along. And then finally, the last category that you need to have a grasp of is the stature type. And so stature types are named after or eponymous with certain typical genera that have that particular stature. So let's take a look at these each one at a time. So Plutioid, which is number one, is named after the genus Pluteus. What do you notice about that, by the way? So look at that number one and compare it to, say, number two and number three. So somebody unmute and shout out. What do you notice about that particular one? How's it different? Is there anybody still out there? I'm going to check. Oh, there you are. I'll give you a hint. Look at the gill attachment. So in other words, how's one different than two and three?
It's freestanding gills.
Yeah, these are free gills, right? So you can see that the gills are not attached to the stipe at all. This one's adnexed. You see this little line here? I think that's supposed to represent that if you break it off, it does the ball and socket thing. And you can see that there's a space between that dotted line right there and the gill. But this is free gill, and the genus Pluteus is one of the categories of fungi that have free gills. All right, two and three are colibioid. The genus Colibia, there's no genus Colibius, is actually pretty rare now in California because of a split that happened. And most of what we used to call Colibia now is in the genus Gymnopus. But those have attached gills. They have moderately robust stature, moderately robust. So it's sort of in between two other statures that I'll talk about in a minute. Number 4 through 6 are omphaloid. And what do you notice about the gills of these three?
Those are decurrent.
That's right. They run down the stipe. So that is decurrent, right? And now a lot of times the Omphalotus and Omphalina both have these sort of depressed centers. So, but not always. So omphaloid just basically means decurrent gills. If you look at all the rest of these, none of the rest have that. The decurrent gills. That's omphaloid. And if you remember from my list, we don't talk about omphaloid too much. Mycinoid and marasmioid are what we refer to here. So these are dainty little things with thin stipes, sometimes a bell-shaped cap, sometimes with adnexed gills, sometimes with adnate gills, doesn't really matter. So these are the little tiny things, and I'll be talking about that a little bit more. And then finally, 9, 10, and 11, almost finally, the tricholomatoid ones. So these are robust. These are heavier duty than the colibioid ones. These right here, or the pluteoid, or the mycinoid. So they're the most robust of all. Now, it doesn't look like they're more robust. It looks like they're just shorter, right? But that's just an artifact of how they were drawn. These 10 tricholomatoid mushrooms can be pretty tall and big, but they have a stipe that is relatively bigger and shorter, bigger around, bigger in diameter, and sort of relatively shorter than you see with colibioid or the mycinoid ones. I don't have pleuritoid as a category. We really only have one type of mushroom that's pleuritoid, and that's pleurotus. Anybody want to know what the
Oyster.
Yeah, the oysters. That's right. Very good. So these are the oyster mushrooms. So if oyster mushrooms come in, I'll figure out where to put them some other time because it'll be the only thing in that category. For the most, there might be something else, but probably not. Okay, so those are the three things: spore color, gill attachment, and stature. And by the way, if you're keen to learn more about mushroom identification, you should get California mushrooms. Hang on. You should get California mushrooms and learn how to use the keys, the identification keys in there. And if you, after some experience with these three things, spore color, stature, and gill attachment, you're more than 50% of the way toward uh, learning how to use the mushroom keys for identification. Okay, so let's talk about the pre-sort groups. By the way, this photograph I just took a couple of weekends ago up at Mendocino Foray. Um, and you can see that we, instead of styrofoam trays, we use paper plates and just wrote the names on there in pen. But this is kind of what you're going to be doing. So that's why I have this slide in there. So is that an attempt at pre-sorting? Maybe I'll take some pictures at the Fungus Fair and do a better job of illustrating that. So, okay. So I've shown you this slide before, but not grayed out. So I'm going to go through each of these categories one at a time, show you some examples, highlight the characteristics that we're looking for. So we're going to start off talking about gilled mushrooms. By the way, my joke is that please don't think that these mushrooms are extracting oxygen out of water. Uh, so somebody down the road thought that that they look like fish gills, and that name has stuck since then. There's actually a jargon term for those blade-like structures that we see on the underside of mushrooms. Here, I'll show you. So these things right here, those are technically lamellae, is the technical term. All right, so here I have three examples of some rusty brown spored species. This is the genus Cortinarius, and Cortinarius is definitely going to be a category on the display tables. It'll be a full tray because there's so many different species of that. It's characterized by having a hefty tricholomatoid stature, so thick stipe, relatively short in stature. It has what you typically they call a rusty brown spore color. You can kind of see that here at the base right there. And Cortinarius is so named because it has a cortina. Cortina is Latin for curtain, and it refers to this cobwebby veil that we can see here, the cobwebby covering of the gills before it opens up. This one over here is the genus Phaeocolybia, and it also has rusty brown spores. Kind of hard to tell with this one because it's young, but that's pretty cool because it has this radicating stipe that goes deep into the soil. And then this is my lovely wife standing in a neighborhood where they cut down a tree and out popped all of these Gymnopolis. This is the big gym, sometimes referred to as that. And we're going to see a lot of those come in. They come in big clusters. And this is one where you can see where the spore color is going to be dropped on the mushrooms that are underneath. But you can see it gets quite prolific. This was single fruiting. That's probably one individual with lots of fruiting bodies popping up out of that stump. All right, let's talk about brown spored. And I have two categories here. I have agaricus, which are the brown spored species that have free gills. And then I'll talk about other browns. We'll look at other brown spored groups. So starting off with agaricus. So again, here we have a young one with a pink gill surface. And if you think about the white button mushroom that you buy in the store, that's an agaricus. And when you cut that open, the gills are not pink. They're sort of a off-white, kind of what we call pallid, kind of a dingy white color. And then if you let those open up, which they do with portobellas, then as the spores mature, it turns the gills that chocolate brown color. And so this is too young. This one's still a little bit too young, but agaricus again has the free gills, And most of them have a membranous partial veil that we see here. Not all of them have that. You can see also with this one that partial veil has left some remnants on the edge of the cap as well. Okay, so other brown spore groups include the Hebeloma and Quinosabe and Inosabe. And again, this is a shadow, but uh, that's not if you just sort of look at that, you might think it's a black spore print, but it turns out that this one has brown spores. You can see the gills here are a little bit on the brown side. There are some mushrooms that'll fool you, like I showed you before. Um, but for the most part, if you see brown on the underside like there or like here, that's probably going to go in the brown spored non-agaricus group. In other words, they're brown spored species that have attached, not free gills. Okay. All right, black spored. So there are a few species that have black spores, a few that you're familiar with, probably. This is arguably one of the most common species that are found in the Bay Area. That's has a nickname of chip cherries, and so these grow in wood chips, as you can see. They're bright red color, and if you look on the underside, you can see the gills are kind of dark. And if you took this one, for example, see how that stem is a little bit bent like that? You might see that, and that's probably because it was growing out from underneath a wood chip and then went straight up so it could line the gills up with gravity. Um, Probably you would see some a little smoky black spore print right on the top of that stipe where the gills have dropped their spores. This is a close relative called Stropharia. Again, has a smoky black spores. This is Satharella, uh, very dark spore print. And again, you might see that on the stipe, which is white, which helps to see that. And then this one over here, this used to be in the inky cap category. Well, it's still referred to as an inky cap. So the gills will turn black as it matures. Some of you may be familiar with a shaggy mane, and shaggy manes have black spores that also are a black gooey mass that contains the spores, and that's black color. Right, so that would go on this table or this category, black spored species. All right, let's talk about pink spored. And remember, when I talk about pink, it's more of a brownish pink, and you can kind of see that here with this Leptonia. So this one and this one with the white gills are young. This one with shaded gills, that's a little bit older specimen. And you can see that brownish pink color starting to show up. Some of you may remember back in the day that your crayon box had a color called flesh. And that was before the company Crayola realized that there were people who had skin color that's different than mine, for example. But they've done away with it. But that's the same color that we see in these spore prints. And there are two main groups that show that color. So this is it used to be called Leptonia. It's now Entoloma. Here's another Entoloma. So both of these have attached gills and pink spores. This is a Pluteus. And if you remember, going back here, remember I talked about the eponymous name. This is a pluteoid stature, free gills, moderately robust. So jumping back ahead, these have pink spores. And if you were able to look underneath there, you would see that the gills are free. They're not attached to the stipe. typically this group, by the way, grows on logs and heavy branches and stuff, but here it's growing on wood chips. And Marilyn, if you notice, that's the trail that goes up um, to the mall front. That's Hensel Hall back there. Uh, so at the San Francisco State campus, that's where were growing.
Oh, I can see that.
Yeah.
Yeah.
So anyway, so those are the pink spored species. And again, sometimes you have to really look carefully at several species specimens. Sometimes you'll see it on the stem or something else underneath to see that pink color. It's a subtle one, and I would be willing to bet that 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 this would be one that I'll have to kind of keep an eye on to make sure that it's in the right place on the tables. Easily mistaken. All right, now we're in the white sport group. In the white sport, there are many, many, many white sport. And so what I've done is broken that into 6 categories. So let's start off with the genus Amanita. So most of you, I'm sure, are familiar with this one here in the middle. It's probably, actually not even probably, I would be willing to bet my eye teeth that this is the most photographed species of mushroom in the world. It's very showy, has the red with white spots. There are other forms. There's a yellow form and there's a white form, but basically most of them are red like this. So this is the genus Amanita. It also includes the most deadly mushroom in the world, the one that's killed more people than anything else, called the death cap. I don't have a photograph of that here. The and then here's one that's been cut open when it was very young. This is a cocora, by the way. Whoop, sorry. Whoop, there we go. So Amanitas are characterized by a couple of main characteristics. One is that they have free gills, more or less. And there are some that'll fool you. So take a look at this one over here on the right. You can see that those gills are attached barely to the stipe. So there's a little bit of attachment there. But the main characteristic, and most of them, it's really clearly free gills. So they're not attached. But there are some species where you see just a little bit of attachment. But the main thing that I want to show you is that they have what's called a universal veil. So now I've mentioned the word veil before, mostly talking about partial veil. And so this one, for example, has a partial veil. All that tissue right there that's covering up the gills will break away and will leave a little ring, usually, not always, but you leave a ring or a zone that's a little bit different where the gill was attached, and sometimes little pieces on the edge of the cap. Um, but that's called a partial veil. The universal veil wraps around the whole thing, and you can see that here with this cocora, and you can see with this. This is a grisette. This grisette, the cap is popping out of that covering that covered up the whole thing when it was young. Now, with most amanitas, as that happens, it leaves patches on the cap. And in the case of Amanita muscaria here, it leaves little small patches. The cocora, which is a really good edible, by the way, but make sure you got the right thing. The cocora leaves a big, thick, felty patch, single patch, instead of a bunch of little patches like the Amanita muscaria. And you can see with this grisette right here, and this may have been because of the weather, there's only a tiny little patch left, and that could fall off or get brushed off. So some of them you don't see it, but they all start out the same way. And then they also leave a cup at the base called a volva. And you can see that very clearly over here on this Amanita gemata here over on the right side. So you can see that very distinct cup shape. And you can imagine that the cup that's left at the base of this cocora is going to be thick and felty. And then you can see the cup that's going to be left at the base of this grisette. Okay, so those are the members of Amanita. So there will be a specific place on the tables for you to put the Amanita species. All right, moving ahead, let's talk about Lepiotaceae. Now, by the way, notice that I put that Lepiotaceae in quotes. That's a family name that has since been subsumed into Agaricus. So, or the family Agaricaceae. So you may think to yourself, well, wait a minute here. Agaricus has the chocolate brown spores, and we're in the white spored category. What's going on? Well, it turns out that these members of what we used to call Lepiotaceae genetically are most closely related to Agaricaceae, such that it's inside of the group of the what we call the clade, the lineage, and so they've been moved over to Agaricaceae. So they're essentially white-spored Agaricus species. That said, the gill attachment will be free, just like on the Agaricus species. So if you see something that has free gills but white spores, not pink spores, not brown spores, but white spores, then it's going to be a Lepiota or a Lepiotaceae in the old sense of the word. Over here on the right side is a pretty classic type. Oftentimes they have this brown center with a white edging, and these can be pretty fragile. So when you're handling those, be really careful with them. Try not to break them. They oftentimes will have this beautiful little persistent annulus. That can be a key characteristic, by the way, if that's present or not. This is called Lepiota procera. This is from the East Coast. We don't get it very often out here, but you can see its resemblance to Agaricus. So that looks a lot like, like a crimini or a portobello or something like that. But again, it has that dark spot, free gills, and white spores. And then this was growing in one of my planters. You may recognize that actually. This is an introduced. I think it's called a Copernopsis now or something like that. But it's yellow, bright yellow, sulfur yellow color, and it typically will grow in planter pots. I probably get asked about this one more than any other thing. It's like this is growing with my rhododendron or with my philodendron or whatever, and it's like that's okay. It's not going to bother anything. It's a decomposer, so it's just eating the rich soil that's in the pots, not the plants. But beautiful thing. And by the way, if you do see these and it comes with a geode, I got dibs on the geode. So okay. All right, moving ahead. So the next group is a single family that has essentially two genera in them, Russula and Lactarius. So Russula, like this Russula silvicola down here, typically has white, sometimes white that stains, sometimes with a little rosy blush, stipe, attached gills, white or cream spores. So some of the spores are slightly pigmented with a little bit of yellow in them. Some of the species have spores that color. Sometimes they're brightly colored, sometimes not. This was I did some work collecting mushrooms in Yellowstone. I actually got paid to do it. Not very much, but I still got paid to go collect mushrooms in Yellowstone. And this was one day's collection of caps from Russula. So you can see quite a range of colors there. Now, the other thing about Russula is that they have specific types of cells called spherocysts. Sphero means ball shaped, cyst means cells. And so when you break the stipe of a Russula, it breaks like chalk. Now, I don't want you to go around breaking a lot of the Russulas. In fact, that's kind of a fun thing to do, to stomp on them or throw them up against a tree or something. But please don't do that during the fungus fair too much. But you'll kind of get a sense for it. Also, the gills tend to be very brittle, so they might even crumble as you pick it up a little bit. But you'll get a sense for that. Lactarius is also has spherocysts. So they also, parts of them, not as much as Russula, but they also, the stem, for example, will break like chalk. But you can see on this illustration here that they have latex in their tissues, and that's where the name Lactarius comes from. So they have a milky juice. Sometimes, like with this one, I think this is Lactarius californicus. It's white. We have red one. We have species that have red latex. We have species that have carrot orange latex. Not in California, but there's one with blue latex that's really cool. But most of them are white. And sometimes they stain. This one stains purple after the latex dries. So it's pretty cool. So that's a category in and of itself. And these are both in the same species, same family, Russulaceae. All right, a tricholomatoid white-spored mushroom. So these are the ones that have white spores, but they're robust, right? So they have a pretty substantial stem. Actually, if you have been out mushrooming much in the past, you might know what these are, what some of these are. So anybody know what any of these four species might be?
Bluets?
Say again.
Bluets?
Yeah, there's a bluet in the lower right corner. Good. And that has white spores. By the way, there's a Cortinarius that looks like that with rusty brown spores. Yeah.
Those white ones aren't Matsutake, are they?
Yeah, that's right. There's Matsutake with my friend Shannon sitting next to him.
What a score.
Yeah, you can smell it, right? It's got that dusty or musty cinnamon smell to it.
Haven't had the pleasure yet.
Yeah. How about this big cluster up here? Anybody know what that is?
That's not honeys again, is it?
Yep, that's a different kind of honey mushroom, and you can you can't really see it here, but that definitely has some spore drops on the mushrooms underneath it. So this is a big cluster of honey mushrooms. And then this was the runner-up for the California state mushroom. Not edible. Anybody?
Jack-o'-lanterns.
Jack-o'-lantern. This is the jack-o'-lantern, glow in the dark, barely, glows in the dark kind of mushroom. And so anyway, looks like it should have orange spores, right? But it has white spores. So watch for that. Okay, so those are the white spored tricholomatoid species. And then we have the white spored colibioid species. So these are not real fragile, but not real robust. And so they include things like this beautiful Lacaria, Amethysteo occidentalis. Again, another beautiful mushroom, the parrot mushroom in the genus Hygrosabe. Getting tired. And then this used to be called Colibia brassicolians, but now it's called Gymnopus brassicolians. So those are the white spore colibioid, and then compare that to the marasmioid and mycinoid species. So these are the real fragile ones. Sometimes they're real tiny, like this one. Those are Douglas fir needles that it's growing on, or like this one. This was abundant at the Mendo 4A. That's the Strobilurus growing on a Douglas fir cone. And then these two are mycenas. So again, you don't need to know that This is a marasmius, that's a strobilurus, and that's a mycena, but they're all small and fragile, and they'll all go in the same place. All right, and then now we'll move over to non-guild, and this will go a little bit faster. So we'll start off talking about the cantharelloid fungi. So this is a cantharellus-like species, including chanterelles. So this is California chanterelle, now our California state mushroom. And then this is a turbinellus. Used to be called gomphis. It's now called turbinellus, but they have similar characteristics, even though they're not related to each other. And that is that instead of having gills like blades, they have ridges. And so here's a close-up of a turbinellus right here. I think it might even be the same one that I took a photograph of. So you can see that they're more like ridges. Frequently, as you can see here and you can see over here, they have a little bit of inter ridge branching that's going on, little connecting between the ridges, but it's definitely not gills. And so those that would be a segment of the one of the tables. The boletes, so these are the ones that have the tubes. Um, Again, see, I think Maria, you were the only one in our group that was at the fungus fair. I don't know if you saw that one. I mean, Mendo. I don't know if you saw that beautiful bolete that this gentleman collected.
Yeah, it was from, he found it in my foray.
On your foray. Oh, yeah, cool. Pretty magnificent specimen. Really weird cap on it, but that's okay. I'm sure it was still delicious. So, so this is the Boletus edulis. And then this is a Swillus down here. This is a Xerocomelus. But they all have one thing in common, and that is that they have tubes. And if you look on the underside, you can kind of see it here. It looks like spongy bottom, but they're essentially the ends of tubes within which the spores are developed. And there's an unfortunate characteristic or unfortunate word that's used to describe a characteristic. They're putrescent, which means that they're basically fleshy. And if you let them sit around too long, they'll turn into a big, massy puddle of goo and maggots. So, so anyway, that's what putrescent means. And if you compare that to the polypores, polypores tend to be tougher. They're often woody or leathery, but they also often have the same characteristic, which is tubes. In fact, that polypores means many pores. So if you look on the underside of this red-belted pine conch, for example, if you look at it with a magnifying glass, you'll see little tiny holes, which are the ends of tubes. This is turkey tail. And then this is false turkey tail, or one of the false turkey tails. It's called the gilled polypore. So it looks like it has gills. These actually evolved separately from other gilled species. But it looks a lot like a turkey tail from the top. So, and it's more closely related to the turkey tails. In fact, it's the same genus as turkey tails than it is to any of the gilled mushrooms. And then this is an artist conch. It's kind of hard to tell scale there, but that's about maybe 2.5 feet across, something like that. And a beautiful woodland scene using a stylus put into the flesh of that. So the artist conch, when you touch that bottom of the mushroom, like up here, it turns brown and it stays brown. So somebody just basically did some beautiful art. This piece actually belongs to David Aurora. All right, then the gastromycetes. So what does gastro mean?
Stomach.
Stomach, right? So these are the ones, whoops, that doesn't belong there. That should be in the previous slide. This is a polypore. Anybody know what that one is? That's funny.
Is that
Oh,
Berkeley?
Dyers?
Yeah, that's the dire. It's a big old Dyer's polypore, isn't it? That's really a beauty. This was at a fungus fair when we used to have it up at Oakland, at the Hall of Science in Oakland Hills. But anyway, that should be that. Forget that's there. These other ones are what we call the stomach mushrooms. So speaking of big mushrooms, here's my friend Alex with his face next to a giant puffball. Uh, this is a hypogeous fungus, which is a mushroom that grows underground. And then this over here is a bird's nest fungus. And these are actually not related to each other. None of these are in the same category necessarily, um, even though they all have the same characteristic, and that is that the spores are produced inside of something. So they don't have gills or tubes or teeth or something like that, where the spores are released into the environment. They are released in other ways, either by being eaten. If they're a hypogeous fungus, they're eaten by rodents. So if you went to the meeting last night, I think you were there, Mel, right? So she talked about having these odors that squirrels and pigs and so on can dig them up and eat them. Or if it's a puffball, basically it either has a opening at the top, like the lycoperdons, or the outside falls apart when it's mature and releases the spores. Dead man's foot does that, or has basically little packets of spores called peridioles that we see in the bird's nest fungi. The gastromycetes also include the stinkhorns and include the earth stars. So those are the gastromycetes. Again a group of fungi that are not related to each other. So that term gastromycetes is a sort of made up group. It's not a real group, um, but they all have that characteristic of the spores being developed inside of something. All right, and then finally, the last category will be everything else. And so that could be the jelly fungi, corals. This is actually not a fungus. That's a slime mold. That's the insect slime. Um, the ascomycetes, like these two, the morels, cup fungi. This is lion's mane, wild lion's mane out in the woods. Or that is actually a fruiting body right there. That's called a crustose fruiting body. So we'll have a category, basically a section on the table for everything that doesn't go into the other categories, right? And then to finish up, and this is a good time to ask me any questions. We talk about the things that don't really fit into anywhere, like these gigantic things right here. I'm not sure what they are. I'm still working on it. But then this is a bolete though. But look at the size of that one. That's a pretty monster bolete, thanks to Photoshop. Check out these. These are little models that were made by a museum in where was that? I think it was in Idaho. That's where I saw this.
Nice.
Anyways, yeah, little uh, cool mushrooms. There's your book, California Mushrooms, with on top of a russula. I'm pretty sure that's what that is, a russula. And then this right here, I got to say it's a little foreshortened. They're not as big as my head. They're only about this big. But the way, I did a selfie with them to make them look bigger. This is a mushroom that grows on southern beach in South America. This was I took this photograph in Tierra del Fuego, um, and that's called Cyteria, and it's really good. It's edible. It has no flavor whatsoever, but still, you could eat it if you wanted to. So, all right. Well, that's my last slide. So let me field any questions you might have.
Well, I have some questions. So, the first one, I guess I'm confused as to what degree to which we have to ID these, because
Oh,
at the beginning you
yeah.
said that they're going to come with a label with the name on.
Oh, the label will have the name of the foray on it. So
Oh,
that,
but not the,
no,
but they're going to be pre-sorted
yeah. So
by,
what I'll need for you to do is to basically be the intermediary between when they show up and the people who are putting the actual names on them. So you'll be putting them in categories.
okay.
And the
And
categories, these are the categories.
okay, okay, got that. And is this presentation going to be available to us so I can watch it again?
Yeah. Right, Maria? Oh, you're muted.
Yeah, yeah. We're gonna upload it to YouTube, Colleen.
Okay, so I
But
can
but
study.
but I think the big idea is to sort guild from a non-guild and kind of like the bigger categories. If
Okay,
if you
okay,
if you know,
You know,
I don't think
again,
yeah.
do your best. I don't have I'm not I don't have high expectations for you to be able to do this perfectly.
thank
And
you.
so if you can figure out what color the spores are, and if you know like the statutes, statures, and if it's free guild or not, that would really help a lot.
Okay, okay,
But don't put too much pressure on yourself to get it perfectly right.
okay,
Yeah. And you'll get a pretty good, you know, as time goes on, especially if I'm around, by all means, come ask me. And some of you know Norm Andresen. I think he's going to be helping out with that as well. Um, but just and the identifiers, if they, you know, if you see one of them walking around, just say, you know, what color spores do you think that is? Or do you think that should be a tricholomatoid or colibioid? Um, and then again, if you put it in the wrong place in these categories, I will fix it. So,
okay,
yeah.
okay.
All right. I hope that was helpful, and I hope that kind of gets you on the track for, you know, going beyond this and kind of being able to put some names on. And again, um, this book right here, the California mushrooms, which I have right there as well, is has some keys in there, dichotomous keys, and in a glossary. So there's a lot of jargon involved, so you can always look things up.
Right, and I have the book,
Yeah, oh,
so,
good. Yeah. Yeah, you should bring it if you have it, although that's not required. It's not something that I'll expect you to use it necessarily. But if you have it and you see something cool and you find out what it is, you can look it up and read about it. And yeah,
okay,
you'll be pretty busy. So the pre-sort is kind of an intense job, actually, uh, in the sense that there's going to be a lot of mushrooms coming in all of a sudden in waves, basically, and getting them out so that there's room for another batch to come in is going to be really helpful for us. Yeah.
And will there be more information sent out about the time and whatnot for these?
oh, yeah.
So yeah, Maria, can you speak to that?
Um, uh, what was the question, Jeffrey?
Oh, just like closer to the date, specific details about when you want volunteers to show up and whatnot.
I think you guys sign up for specific slots,
Oh, I think okay.
so you guys will get a reminder about what time
Right.
that you sign
Perfect.
up for.
Very good.
I
So
didn't.
the volunteer coordinator's name is Elizabeth and Simonson. Is that right?
Yeah, that's correct.
And so she is going to be sending out periodic updates and reminders. She's great. Please say thank you to her because she's doing an amazing job.
And thank you for the great presentation.
Oh,
Yeah,
thank you, Jeff.
Yes, thank you.
thank you, JR.
Welcome. All right, everybody. Take care. Stay dry.
Okay.
Pray for rain at the same time.